Good morning, everyone. It's my great pleasure to give this presentation together with my colleague, Joe Ecker. We are going to introduce the single-cell epigenome analysis of the mouse and human brain in the context of the brain cell atlas, where we truly believe that this atlas will help us understand not only the composition of the mammalian brain structures, but also understanding how that cell identity and the gene regulatory program is encoded in the DNA sequence. Let me start with a very simple idea that everyone in this room agrees on, and that has a profound consequence for how we study the brain. Every neuron, every glial cell in the glial cell in the brain carries essentially the same genome. What makes cortical pyramidal neuron different from a striatal median sparing neuron is not the DNA sequence itself, but how that genome is regulated, what parts are accessible, what parts are methylated, and how regulatory elements are wired to their target genes. In other words, cell identity in the brain is fundamentally an epigenomic phenomena. If you think about the genome as the text of a book, then the epigenome is like the bookmarks telling the cell which pages to read and when. The epigenome is taking the form of open chromatin, DNA methylation, histone modifications, as well as the 3D quantum structure. It should be noted that while every cell in the body shares the same genome, their epigenome varies depending on the cell type, developmental stages, and environmental context. For a long time, neuroscience has relied on gene expression as the primary molecular descriptors of cell type. That has been incredibly powerful, but expression alone does not tell why a gene is on in one cell and off in another, nor does it tell us how non-coding genetic variations influences disease risk. This is where epigenomics become essential. It provides a mechanistic layer between DNA sequence and cell function. One of the key motivations behind this work is that gene regulation operates across multiple genomic scales. At the smallest scale, transcription factor motifs and single base changes matter. At intermediate scales, enhancers, silencers, and promoters interact. And at largest scales, quantum loops, domains, and compartments determine which regulatory element can even see which genes. The brain is particularly challenging in this regard because these regulatory programs are highly cell type specific and region specific. What we set out to do was to build single cell epigenomic analysis that span all of these scales, from base resolution, DNA methylation, to chromatin accessibility, to 3D quantum organization across the majority of cell types of the brain. We began in the mouse because it allows systematic sampling, genetic perturbation, and experimental follow-up. In 2023, we published a set of large-scale single-cell epigenomic analysis of the adult mouse brain in Nature. These datasets provided hundreds of thousands of individual nuclei across essentially all major brain regions, profiled using single nucleus methylation sequencing and chromatin accessibility approaches. What did we learn? First, epigenomic data robustly recapitulate known neuronal and glial cell types, and in many cases resolve finer distinction than transcriptomics alone. Closely related neuron types that are transcriptionally similar often show striking differences in DNA methylation and chromatin accessibility at key regulatory loci. Second, we generated a comprehensive catalog of candidate cis regulatory elements active in specific brain cell types. Many of these elements are completely invisible in bulk tissues and were not present in the previous reference dataset. This catalog immediately became useful for interpreting non-coding variations and for understanding how regulatory programs differ across the brain. But cataloging regulatory element is only the first step. The deeper question is, how do these elements work together to specify cell identity? To address this, we integrated multiple modalities at single cell resolution: DNA methylation, chromatin accessibility, gene expression, and chromatin conformation. One example from the Nature studies involved cortical-thalamic versus cortical neurons. These cell types express overlapping sets of genes, but their regulatory logic is distinct. By combining accessibility and methylation data, we could identify cell type-specific enhancers. By integrating 3D genome context, we could link those enhancers to their target genes, and by motif analysis, we could infer the transcription factor likely driving each program. This allows us to build cell type specific regulatory networks to predict how perturbing a transcription factor or enhancer would affect downstream gene expression, something expression data alone cannot do. I'll turn this to my colleague, Joe.
I want to give a couple of examples of the use of these resources. So a major challenge in neuroscience is connecting the molecular identity of those cells to circuit function. And to address this in the BICAN, in the BICCN project, we developed a method called epiretroseq in collaboration with Ed Calloway, which combines retrograde tracing, viral tracing with single-cell DNA methylation profiling. This was also part of the package in 2023. We profiled over 33,000 neurons whose projection targets were known, covering more than 200 combinations of source and target across the brain. So what this allowed us to do was to answer questions like, how do neurons with the same projection pattern do they share epigenomic signatures? And the answer was yes, but with some important nuances. Okay, for example, cortical-thalamic neurons projecting to different thalamic nuclei showed subtle but consistent differences in DNA methylation at the regulatory elements that control exon guidance and synaptic genes. And this really demonstrated that projection identity is encoded, at least in part, in the epigenome. And epigenomic profiling can bridge the molecular taxonomy now with circuit architecture. And if we can identify these same features in the human brain, we may be able to predict where those neurons are projecting, and hence obviously can't carry out experiments in humans, but that information can be used to look at, for example, disease circumstances where projections may be altered. Another example is the use of this map to look at aging in the mouse brain. So this was a map, these maps were made from P56, postnatal P56 mouse brains. And now that's work in press from Bing's lab and our lab, and also on the Barr archive, we examined how the epigenome changes with age at single cell resolution. And so one key finding of this is that aging across the brain in different cell types is not uniform. So some neuronal populations show widespread loss of DNA methylation, for example, or open chromatin at regulatory elements, while others are remarkably stable. And certain glial cell types show pronounced epigenomic remodeling linked to inflammatory pathways. So these results suggest that vulnerability in aging and aging-related disease is encoded in cell type-specific regulatory programs, not just in global changes across the brain.
As these data sets grow, it became clear that we needed a census-level approach. This culminated in the brain cell census, published in Science in 2023. This effort integrated over 1 million single cell across modalities to produce a unified taxonomy of brain cell types and a massive catalog of cell type specific regulatory element. One striking result was that the majority of the brain regulatory element are cell type specific and were previously unannotated. This immediately explains why most genome-wide associating study risk variants fall in non-coding regions because we simply lack the right regulatory maps. The brain cell census transformed epigenome analysis from descriptive resources into imperative frameworks for disease genetics. For example, we were able to use this data to train a machine learning model using the framework that was recently reported called Borzoi. We use DNA sequence as the input, train these models using the epigenome datasets that was just described above, and the result was striking. We were able to show that the models predict transcription and epigenetic properties quite accurately, as you can demonstrate, as we can see here from the predicted observation and the true observation. We can also show that with more modalities, such as using histone modification on top of RNA and chromatin accessibility data, we gained in predictive power. Of course, mouse models are only part of the story. In this study, we extended the single-cell epigenomics to the human brain, combining DNA methylation profiling with single-cell chromatin conformation capture. In another study, we mapped chromatin accessibility and single-cell resolution for nearly 1 million brain cells isolated from 42 brain regions collected from 3 phenotypically normal donors. This effort resulted in one of the first single-cell analysis of the human brain, uncovering over 500,000 brain cell-specific regulatory element across 170 distinct brain cell types. I'll turn to Joe.
As being described, we extended the studies from mouse to human brain, initially focusing on the cortical areas. And we combined DNA methylation and chromatin cell contact information to begin to build a 3D genome architecture of human neurons and glia across a variety of brain regions in that initial study published in Science in 2023 is being described. An important finding was that human neurons exhibit distinct compartments and domain structures compared to the mouse, particularly at loci involved in synaptic function and neural development. And these differences may underlie human-specific aspects of cognition and disease susceptibility. And the data set also allows species comparison, revealing which regulatory programs are conserved and which ones are unique to humans.
At this point, we had extremely rich data set, but we want to go one step further. Could we learn the regulatory grammar encoded in DNA sequence itself? In this 2023 science study, we trained deep learning models on brain epigenomic data set to predict cell type specific chromatin accessibility directly from DNA sequence. These models achieve high accuracy across diverse brain cell types, indicating that they have value for us to understand information encoded in the DNA. As an example, we focused on a brain region type of cell called microglia cells, and one region in this type of cell harbors 2 risk variants for the late onset of Alzheimer's disease. We can perform in silico mutagenesis to predict that one of these variants, upon mutations, could lead to a change in chromatin accessibility, while the other variants have no such impact. This prediction is validated through independent CRISPR editing experiment conducted in Dr. Ying Shen's lab at UC San Francisco. These studies exemplify how combining single-cell epigenomic analysis with deep learning approaches can enhance our understanding of the non-coding genome. By integrating high-resolution data with advanced computational models, we can predict the impact of genetic variations on gene regulation within specific cell types, paving the way for novel therapeutic strategies.
To be truly useful, for these machine learning models are going to require extensive human brain sampling across developmental stages and across anatomical regions. And so our current UM1 project involves sampling across 3 different developmental stages, and including male and female, and across 71 regions of the brain that include 125 distinct regions of the brain shown on the right here. Next slide. So many of the methods that we're using here were developed in our laboratories, the SNM3C-seq or methyl high C in my lab and Bing's lab, and the pair tag approach. This slide shows the SNM3C-seq, which captures DNA methylation and chromatin conformation in a single assay and allows the different scales to be captured, the scales of the genome that Bing described, to be captured in a single assay from base pairs up to domains. Next slide. The other method that's used in our UM1 that's carried out in Bing's lab is a droplet pair tag assay, which Bing's lab developed, which allows you to capture, really for the first time, multiple chromatin modifications and RNA sequencing from a single nucleus. And that provides very powerful combination of multi-omic assays to interrogate brain cell identity. Next slide. So in this next phase of the project that's really been led by Ed Lean, all of the groups, the UM1 centers, have begun focusing on basal ganglia, which is shown here. Seven regions of the basal ganglia which are being analyzed, and the domains indicated on the slide, on the next slide. Here is some of the sample data that's been produced. This is just from the SNM3C sequence data, and you can see the complexity of these various cell types across the different regions of the basal ganglia. Next slide. So just to summarize what our center is doing, the take-home messages are that first, single-cell epigenomic profiling can reveal regulatory elements that define brain cell identity. Integrating the different modalities that I described allows us to infer regulatory logic, not just classifying the cells. Third, these atlases provide a missing link between the non-coding genetic variants and disease-relevant cell types in the brain. And finally, the computational models that Bing described, trained on epigenomic data, turn these descriptive atlases into predictive tools, enabling mechanistic insight and experimental validation can be pursued. Next slide. And so our center involves a number of laboratories: the Shu Lab at UC Irvine, the Children's Hospital of Orange County, Marga Behren's lab, our core facilities, Bing Ren's laboratory, both at UCSD and New York Genome Center now, my group, and for data display, Washington University, St. Louis has been essential in this analysis. And we thank the NIH for the funding of our center.